The Use of Sequencing Technologies in the Ebola Epidemic by Henry Deng, Alicia Hale, Rachel Smith, Dalton Tucker, and Minji Zdeb. In 1976, an outbreak of a mysterious disease simultaneously struck Nazara, a town in South Sudan, and Yambuku, a small village in the Democratic Republic of Congo. 602 people were afflicted by this disease, and of those 602 people, a total of 431 people died. This represents a mortality rate of 72%. Today, the disease has become known as the Ebola virus disease, or more commonly referred to as simply Ebola. As the name suggests, Ebola is an infectious viral disease, causing severe hemorrhagic fevers and is oftentimes fatal, especially if left untreated. Mortality rates from outbreaks of this disease have even been documented to reach as high as 90% in a sizable population. Although researchers have identified several species of viruses that cause Ebola, 3 species in particular have been associated with large outbreaks in Africa: the Bundibujo, Zaire, and Sudan Ebola viruses. There are also 2 other species of Ebola that have been discovered, and they are the Thai forest Ebola virus and the Reston Ebola virus, the latter having only caused disease in non-human primates. These viruses are believed to have been introduced into the human population through the exposure to the bodily fluid of infected animals. This includes chimpanzees, gorillas, fruit bats, and antelopes. Once inside a human host, the virus can spread to other humans through direct contact with bodily fluids of an infected person, or with any surfaces that have been contaminated with these fluids. Once a person has become infected, symptoms usually begin with a sudden onset of fever, fatigue, muscle pain, headache, and sore throat, followed by more severe symptoms such as vomiting, diarrhea, and sometimes internal and external bleeding. Currently, there is no cure for Ebola, and treatments usually focus on supportive care and treatment of specific symptoms. This can include providing intravenous fluids, otherwise known as an IV, balancing electrolytes, maintaining a consistent oxygen level, maintaining a consistent blood pressure, and treating other infections quickly if they do occur. Although major outbreaks have been largely isolated to Africa, especially in countries with poor healthcare systems, lack of human and structural resources, and civil conflicts, Ebola is considered to be a serious threat to the international community. The current Ebola outbreak in Western Africa, with the first case being documented in March of 2014, is the largest and most complex of all outbreaks to date, with the number of cases and death toll exceeding all previous outbreaks combined. The scope of the current outbreak has prompted the World Health Organization Director General to declare a public health emergency of international concern on August 8, 2014. Considering our increasingly globalized world, the Ebola crisis has also highlighted the need for technologies that can be used to track transmission chains and viral evolution, especially for those that can be used to track diseases in remote areas during outbreaks. One such technology that has recently emerged is the MinION, a type of nanopore sequencing device designed by Oxford Nanopore Technologies. Compared to other traditional sequencing devices, which are large, expensive, fragile, and difficult to transport, the MinION, which weighs less than 100 grams, is much more suitable for use in remote locations where infrastructural support is limited. The MinION works by measuring disruptions in ionic flow as a single strand of DNA is passed through a protein nanopore at a rate of 30 bases per second. Each nucleotide base of the DNA causes a characteristic disruption in ionic flow, which is detected by the device and recorded on a laptop. In studies conducted at the Rocky Mountains Laboratory and NIH Field Laboratory, it was shown that the MinION could successfully and accurately sequence complete virus genomes from previous outbreaks. In 2015, the MinION was also tested in an actual outbreak situation in Guinea. During this outbreak of Ebola, 142 Ebola viruses were sampled and sequenced over a period of 6 months, with each sample taking an average of 2 days to sequence. The rapid rate at which the device was able to successfully sequence the Ebola virus genomes allowed researchers to monitor the outbreak in real time. But despite its advantages, there are still several limitations that have prevented the widespread adoption of the MinION in the remote and resource-limited areas. First, the PCR thermocyclers that are used to provide the MinION with amplified DNA relies on a continuous and reliable source of power. Second, the analysis of genome sequences requires reliable internet access, which is often an issue in remote locations. And third, since the MinION can only sequence amplified DNA sequences, it is unable to sequence the genomes of unknown pathogens. However, even considering these limitations, nanopore sequencing technology, such as the MinION, represents a major step towards our ability to efficiently monitor and track emerging infectious diseases in real time. Using prime synthesis DNA sequencing methods, complementary DNA was able to be made simply by the fractionation of different sizes of DNA fragments using electrophoresis.